All right, we're close to 30 and it's 8:01 and we're gonna make sure we give Eddie his due time. So if it's okay, I will get started. Welcome everybody. I don't know what month we're in. I think it's June 2025. But welcome to another High Rounds. We have one of our fellows who I thought was graduating, but he was like, no, I'm only in my third year. That's how long Eddie feels like he's been doing the good work. Um, so he's from Oakland, California, graduated from Case Western Reserve University School of Medicine, and did his residency here at UCSD. He's a second year, well, about to be third year infectious diseases fellow on the research track, and his research focuses on preventing mosquito and tick-borne infectious diseases. His hobbies are running, backpacking, and hiking. A lot of good stuff that he likes to do. So maybe we'll get into that later. But this is a departure from what we often do. But I do like to remember people that High Round stands for HIV and global health rounds. So we're taking a little turn to global health, and hopefully this will be really interesting for all of us. So with that, Eddie, I'll turn it over to you, and I'll keep an eye on the chat for you and give you a reminder when we're close to the time.
Cool. Thank you, Jill. Appreciate it. Thanks for the introduction. Um, okay, so let's get into it. So I wanted to start this presentation off by saying that, mosquito-borne illnesses are on the rise. So this is a global issue, as Jill mentioned. So the WHO, for example, they reported eightfold increase in global dengue incidence in the last 20 years. So this is a result of increased travel, urbanization, rising insecticide resistance, and rising climate temperature. So this is a animated graphic that shows how the Aedes aegypti habitat is expected to change if climate temperature continues to rise as it at its current rate. So you'll see that it'll creep into some northern countries, US, Europe, East Asia that it previously hadn't been able to go to because it had been too cold. So again, this is a global issue, warrants a multifaceted preventative approach, and I wanted to talk about that today. So the objectives of this presentation it's going to be like kind of a whirlwind tour through mosquito control approaches and with a focus on recent or novel approaches and advances. We'll focus on 5 different areas. We'll talk about chemical control, barriers, environmental control, genetic technology, and then also biologic control as well. A lot of what I'll be talking about is based on armchair research, but also kind of bench work that I've been doing during my research year. But if you've been in areas that have been particularly plagued with mosquito-borne diseases, and have like seen, up close how these diseases are practically managed or prevented, please speak up, because I think we'd all really like to hear. Um, I have never ever had to disclose anything for my presentations before, but this time I do have to say that as part of my research, I do have a patent pending on some mosquito-proof textiles, as part of my lab work. So the first facet of mosquito control that I wanted to talk about is chemical control. I chose this as the first thing to talk about, mostly because the issues with chemical control help us understand why a multidisciplinary approach is so important to fighting this battle. There's lots of significant limitations. Um, so for this portion of the talk, I will only be talking about insecticides. So I won't be talking about chemicals that are pure repellents like DEET, but most of the same concerns exist for DEET as they do for other insecticides. So there are 4 major classes of insecticides used today. Organochlorines are they're mostly out of favor. They're completely banned for this for this indication in the US, but they're still used elsewhere. All of these four major classes of insecticides are neurotoxic. So they either affect the acetylcholinesterase, voltage-gated sodium channels, or GABA receptors. So they either cause coma or death, either of which kind of lead to, I'm sorry, they either cause coma or paralysis, either of which lead to death. Included a picture of a chrysanthemum here because pyrethroids are analogs of pyrethrum, which is extracted from these flowers. So these flowers have kind of long been known to have insecticidal properties. And we got permethrin basically from these flowers. Insect growth regulators are another broad category of insecticides. They target larval forms of mosquitoes. And as a reminder, part of the life cycle is the larvae live in the water, and once they become adults, they emerge from the water and start flying around. The chitin synthesis inhibitors prevent hardening of the cuticle. So basically, they're not allowed. They can't fly if the cuticle isn't hardened. So they can't fly out of the water, which is the basically the final stage of metamorphosis. And there's multiple different insect hormone analogs, which disrupt different steps of the of metamorphosis as well. There are multiple ways that these chemicals have been employed. The WHO stresses two in particular. Number one is indoor residual spraying, which is kind of spraying the walls and surfaces of the home with long-acting insecticides. Mosquitoes like to kind of rest indoors, and if they do so here, they will most likely die. And then the WHO also recommends nets. We'll talk about that a little bit later with the barrier methods. Um, but yeah, I mean, we spray textiles, all sorts of textiles with insecticides, not just bed nets and clothing, meshes, things like that as well. Larviciding is a mosquito control strategy that targets mosquito larvae. So this, I mean, often involves the insect growth regulators that I mentioned before. Basically, we spray these insecticides into mosquito breeding grounds like stagnant ponds and puddles and rice fields. And this is a particularly effective strategy in urban environments where we know they're breeding, basically, where we know the breeding site specifically. Another common strategy is fogging or space spraying. It's basically involves basically just widely dispersing these insecticides into the air as a fine mist. And this is the only reactive strategy of these four, and it's done during outbreaks. So if there's just tons of mosquitoes out, these communities will spray these insecticides in the air, um, hoping to kill the mosquitoes. There are lots of issues with the use of these chemicals. So mosquitoes have developed resistance because of selective pressure on these like very specific neurologic targets. So in the 2021 World Malaria Report, 78 out of 88 malaria endemic countries reported resistance to neurotoxic insecticides. And a lot of these resistance mechanisms have to do with pyrethroids because those are the most widely used insecticides used in bed nets. And I wanted to talk about a few of these mechanisms. The first is increased sequestration and metabolism. So sequestration involves basically attaching these insecticides to proteins and activating them, or transporting these insecticides to kind of fat bodies or gut tissues or vacuoles and just basically away from the nervous system, so they can't work. Metabolism. Cytochrome P450s are very effective at metabolizing these insecticides and in activating them. There are kind of, I mean, target site modifications are another important and common resistance mechanism. Cuticle thickening is another one. So increasing the thickness means it basically blocks the insecticides from entering the body. This is a model of resistance over time based on genotype data for those those genes and alleles that we've identified as resistance carrying. And so you can see that over the past, from 2005 to 2017, there's been a dramatic increase in resistance, specifically in West Africa. Um, and then the last resistance mechanism that I wanted to talk about is behavioral avoidance. So we'll talk about this a little bit later, but these mosquitoes will basically start avoiding those bodies of water that have received larvicidal treatments. They just know to avoid them at some point. So, in addition to genetic resistance mechanisms, and behavioral changes, insecticides are limited by non-target toxicity. So, in humans, they can cause skin and eye irritation, particularly if, they're worn as a clothing item. Um, they can be systemically absorbed as well. The systemic absorption increases with heat and with sunscreen. Um, and systemic absorption has been linked to neurotoxicity, even with DEET. And DDT, which isn't allowed in the US anymore, has been specifically linked to androgen and estrogen disruptions and non-Hodgkin's lymphoma. These also affect non-human animals. So DDT, for example, can lead to significant declines in multiple bird species by disrupting the calcium pathway, leading to really fragile eggshells. These chemicals are very environmentally persistent. There's just a general sense that they're causing negative effects on the environment because they're just always there now. But the consequences, I think, are generally unknown. So with that being said, I wanted to move on towards some more recent approaches and advances. To tackle the behavioral part of this, auto-dissemination ovitraps traps have been developed. So this is basically a novel way of using mosquitoes to spread the insecticides themselves to all their kind of secret breeding areas. So basically, mosquitoes are attracted to a hub with standing water that is treated with a larvicide, like an insect growth regulator or something like that. And then when they lay their eggs, they get dusted with these small doses of the larvicide. They fly off and they visit multiple breeding grounds and basically poison those wells as well. So it's been found to be effective at reducing Aedes aegypti populations in field studies in Brazil, Tanzania, and the Philippines. So it's a really cool method. Insecticide rotation is now widely recommended in order to slow resistance. So if we look at this graph, the y-axis is log 10 scale. That's a scale at least. So the value of 0 corresponds to 100% frequency of the resistance allele. So frequently switching out the insecticides, like every 6 to 12 months, for example, this is the right graph. You can see that it leads to a longer time before the population is considered resistant. So it gives you some time basically, to figure out some other techniques to fighting mosquitoes. Um, and then PBO is another really important recent development. So this is not an insecticide, but a cytochrome P450 inhibitor that decreases metabolism of pyrethroids. Dual active formulations are also commonly employed now. So basically, don't just do pyrethroid, but kind of add another agent on top of that to make it hard for mosquitoes to survive and develop resistance. There's new insecticides out there. So chlorphenipur is one that decreases mitochondrial ATP production. And I wanted to talk about another drug I found interesting. It's a drug that works on glutamate gated chloron ion channels. I don't know. I mean, can anyone guess what this? So this is a medication that we already use in the medical community today frequently, but for different indications. Does anyone know what medication I'm thinking of that is now being developed as a new insecticide? It's kind of a guess what I'm thinking question. But it's ivermectin. So ivermectin is helpful. So, in the early 2000s, countries were using ivermectin in mass drug administration to control river blindness and lymphatic filariasis. And they incidentally found out that during those trials, mosquito densities dropped significantly. So pretty soon, controlled lab studies and controlled community trials confirmed this, that mosquitoes had reduced survival when exposed to ivermectin. So this is from the Ivermol trial in Kenya, published in 2018. So this is a trial that looked at patients who were diagnosed with malaria, and they only included patients diagnosed with malaria. And these patients were given ivermectin in addition to their anti-malarial treatment. And then mosquitoes were allowed to feed on these individuals at different time points after treatment. And the investigators saw that the mosquito's median survival, which is the y-axis here, was lower in those patients who had been given ivermectin. So that's kind of, and that's seen in the red and green lines here. So this was most noticeable in the first 7 days. And ivermectin was fairly well tolerated too. So it's like, really good option. I mean, maybe in the future, we'll just be giving everyone with malaria ivermectin. So that would kind of maybe reduce, maybe protect the community, definitely protect their family members a little bit more. So it's a cool option. West Nile virus is the most common mosquito-borne illness in the United States. It's primarily transmitted by Culex mosquitoes. Culex mosquitoes have West Nile has these like amplifying hosts, these birds. They're basically songbirds. And these are amplifying hosts are hosts that develop prolonged periods of high viremia. And I mentioned this important part of the West Nile virus life cycle because there are current experiments going on in Colorado and California that are looking at feeding birds ivermectin-coated seeds during the summer months to decrease mosquito survival and basically reduce the transmission of West Nile. So these birds, there's these you know these birds consume ivermectin seeds, little scattered areas throughout California and Colorado. Mosquitoes bite them, maybe pick up some West Nile virus, but then just live a much shorter period of time and reduce incidence potentially. Um, it's a really kind of creative and cool strategy to combat West Nile. And it's a medication that we already know is relatively safe in humans.
Eddie, sorry. Dr. Blanchard just had a quick question. I
Yeah.
don't know if you answered it, but did those patients get bit less? So I think it was on the,
I'm sorry, can you say it again?
did the patients get bit less? I'm actually not sure what she was.
No, so for the patients who got treated, the malaria patients who got treated with ivermectin,
Oh yeah.
you, did they
Yeah.
get bit less?
They got it. No, well, it was controlled. So they got a certain number of bites.
Hmm.
They made sure that I don't know if it was harder for the mosquitoes to bite these patients. But they
Well, I'm
made sure
just
that,
wondering, yeah, I'm just wondering, for all the stuff we spray on ourselves, I'm wondering if there was something about their skin that
yeah.
protected them as I go off to Minnesota next week.
I don't think they were investigating. I think they really just wanted these mosquitoes to bite them the same amount for each one of these patients at certain time points.
Got it.
I'm not sure if they looked at anything else behind that. So
Okay, thank you.
yeah.
And then there's another question from Dr. Littles. I don't know why I'm doctoring everyone today. This is casual. How safe is ivermectin in the birds? Do they do they get
So they're
harmed?
looking at that. These are really recent studies. And so I don't think they quite know. I actually didn't see a full paper about this yet.
Okay,
That's a good question.
that's
Yeah.
all I got.
Um, Yeah, so keep asking questions throughout. That's perfect. So that was, that's a quick run through chemical control. Next are barriers. So starting with, the probably the most incredible life-saving measure, these bed nets that have prevented 1.5 billion malaria cases, more than 7 million lives saved. Bed nets are generally sprayed with insecticide or repellent. Previously, it was just with the pyrethroid, but now, in response to rising insecticide resistance, we're just switching it up a little bit. So, in recent years, we're combining it with PBO, which again is that cytochrome P450 inhibitor, or we're just adding another agent on board. And that's seen in this graph here, where the ITNs on the y-axis is insecticide-treated nets, and then x-axis is time. So we've just had to modify our approaches with the nets at least. I think that's part's definitely helpful, but still somewhat limited by behavioral changes. I unfortunately didn't have access to this article, but it was published in 2012 in the Journal of Infectious Disease. It showed that 1 to 3 years after the deployment of long-lasting insecticide-treated mosquito nets in 2 villages in Benin, Anopheles basically just significantly changed their behavior. So they more frequently bit outdoors as opposed to indoors. And it was just later in the morning as well. So it's generally 5 a.m. compared to 2 to 3 a.m. So that was just in 1 to 3 years. So to me, it's like a fairly quick adaptive response to bed nets and the insecticides on. Clothing is also an important barrier method. So clothing on the market right now is generally covered with pyrethroids. Not only does it have some toxicity, but it comes off in the wash. So it'll just lose efficacy after just a few washes. And to maintain efficacy, you basically have to reapply insecticides frequently. Um, as an aside, while washing damages the chemical effects, the high heat drying, if you were to have a dryer, it improves the barrier effect. So for obvious reasons, it tightens the weave structure. So it actually enhances the barrier effect. I think covering the body completely with long sleeves and pants can certainly be helpful, but it's not completely helpful. So this image shows where clothing clings to bodies and in men. Um, and these are just high risk areas for mosquito bites because these are areas of the body where the weave is stretched and the fabric pores are bigger. So the proboscis can get through and bite the skin more easily. And there's also just less space between the fabric and skin in these areas. Um, so as part of Dr. Omar Akbari's lab, I've been investigating insecticide-free fabrics that completely block mosquito bites. And through this project, I learned that, you know, not only do we need to make sure that mosquitoes are not blood feeding, we have to make sure that they're not biting. Um, they don't necessarily need to be able to find blood vessels in order to transmit these diseases. So after tons of experiments, we found kind of multiple comfortable insecticide-free fabrics that were bite-proof against the three main genera of vector-borne diseases. So we have Anopheles, Aedes, and Culex. And so this is a video of one of those fabrics and experiments, and you can see the mosquitoes. There's a mouse on the other side of this. They're attracted to the mouse. They're looking for a spot in the fabric to kind of stick their proboscis through, but they've been unsuccessful so far. Other barrier methods include just sealing up housing structures, windows and door screens. Eaves tubes are a recent big deal. This is a diagram produced by the Seattle Times, which is fitting because the Gates Foundation has been heavily invested in eave tubes. So basically, traditionally eaves are open, but now we're adding tubes into these eaves that have an electrostatic netting that's impregnated with insecticide. So it blocks mosquitoes, also kills them. Pretty effective. Um, Environmental management. I guess I only have like 20 more minutes, so I'll go quick up through this. But so basically, these are the kind of the key interventions that the WHO has laid out to disrupt mosquito breeding grounds and reduce population numbers. So the biggest one is, standing water is not good. So we need to prevent standing water formation. That involves a lot of upfront money. We have to kind of modify landscapes if possible, kind of fill in ponds or ditches, and then also have good irrigation systems. So this is a graph looking at the abundance of two different Culex species in California. Um, the abundance is on the y-axis, x-axis is time, and then the different color uh, lines have to do with how much irrigation those areas receive. And so the lighter green is low irrigation, the darker green or black is high irrigation. So obviously it looks like that there's a correlation between less irrigation means fewer Culex mosquitoes transmitting West Nile, for example. So we don't need flooding systems for rice fields. We can use intermittent irrigation or drip systems, and they appear to work just as well with the reduced incidence of mosquito-borne illnesses. The WHO also recommends, securely covering water storage containers, and then also removing and managing accidental containers. So this is a often forgotten um, breeding grounds for mosquitoes. So forgotten pots, vehicle tires, broken coconuts. These are all things that can breed mosquitoes, and covering them and removing them is very helpful. And then the last thing that the WHO recommends is like safely storing and removing domestic waste. I've talked about this a little bit, but there's limitations to the environmental reform. I think it just costs a bunch of money, particularly upfront. Heavy equipment is needed to kind of make these temporary or long-lasting land changes and to make new irrigation systems. A lot of communities can't do that. Off-target effects, including, if we fill in ponds, what other organisms might be affected? And then there's just a huge coordinated effort that I just can't even imagine trying to figure out. It just requires buy-in from an organization between the different community and different community members and agencies. It just seems very, very difficult, but not impossible. And then land use conflicts may arise from that. So this is where a ton of exciting things are coming out from Kume to cover in this lecture. I'll mention two strategies though, both from Dr. Akbari's lab. So the first, in my mind, this is the biggest deal. So this is precision guided sterile insect technique. It was created in 2018 and is a form of population suppression. It's been successful in mosquitoes and flies, and we're trying to get it to work in sandflies and ticks. Although I guess ticks aren't technically insects, but I think the same process applies. So let's talk about how it works. So basically through micro injection into mosquito eggs, so that's seen here. These are mosquito eggs. Basically, the first step is to create two stable homozygous lines of Aedes aegypti mosquitoes. So one contains Cas9, which cuts DNA, and then one contains kind of multiple sex-specific guide RNAs. So one guide RNA targets myosin heavy chain, which is just essential for flight in females, but not in males. And then another one targets beta tubulin, which is essential for spermatogenesis in males. And so two lines are crossed. The guide RNA binds to Cas9. You can see that in this picture here, and then guides it to the target genes. Again, the target genes are in females, it's myosin heavy chain, essential for flight, and then in males, it's beta tubulin, which is essential for sperm production. And the Cas9 protein causes a double-stranded break at the target gene, and then non-homologous end joining leads to disrupted genes. And so the resulting offspring contain the guide RNA Cas9 complex. The females have impaired myosin heavy chain. They are flightless. They can't leave the water surface. They can't blood feed. They're basically non-viable. And then on the other hand, the males can't reproduce. They're sterile. So basically, so because the females essentially die, this technique skews the sex ratio towards males. And so these males, which males don't blood feed. So I should have said that up front. So it's just females that blood feed. So the males will compete with wild type males for mates. And there's not a fitness cost to this. So they're often successful. So the result is a slow reduction of the population. Um, and because the males are sterile, this genetic technology is not is not widely propagated. The only way that it persists in the environment is if the mosquito population, if the eggs are consistently released into the population, as seen on this right graph. So it's in this sense, it's thought to be safer, right? So we just create some sterile males that outcompete the wild type males. We can decrease the population steadily, and we just continue to release these eggs into the environment. I won't go into this right now. Actually, I'll skip it right now. But gene drive is a completely different, not completely different, but it's a different mechanism of controlling population or suppressing population in mosquitoes. It was if you uh, Gates had that recent Netflix like short series. I think it was highlighted in one of those episodes. But anyway, so PGSIT and gene drives are different um, genetic technologies that have the promise of potentially kind of decreasing mosquito population, but they have not been used in kind of true field tests quite yet. We don't need to just genetically modify mosquitoes, though. We can genetically modify skin bacteria that attract mosquitoes to us. So as background, mosquito attraction relies on olfaction. So they sense CO2, and that sense is strongest when the CO2 synergizes with other compounds that are generally created by skin bacteria. The most, the most important synergistic compound is lactate. So in this study, the Akbari lab treated two populations of Staph epi and then Corynebacterium, which are common skin commensals, and deleted their lactate dehydrogenase. So they couldn't create lactate anymore, and then engrafted these populations onto mice, and this caused significantly decreased attraction and feeding. This is a sampling of the data looking at differences of mosquito attractiveness on the y-axis with wild-type Corynebacterium, which is in purple, and then the knockout is in red. So knockout meaning that they don't have lactate dehydrogenase, they can't produce lactate, which again synergizes with CO2 and is a big attractant. And you can see that there was no significant difference on day one, but for the next 14 days, these mosquitoes were not attracted to these mice nearly with these like modified skin bacteria compared to the wild type skin bacteria. 14 days is, it's a long time to repel mosquitoes. DEET lasts for 4 to 8 hours before you have to reapply it because it evaporates otherwise. So this is like a significant improvement in repellency. And one of the natural next questions would be, okay, so what are the next steps? How could this practically be employed? So what we could do is, we could do what they're already starting to do in dermatology, which is bacteriotherapy. So, I think this is just a cool little thing to mention because this is just a whirlwind tour anyway. It turns out that eczema is frequently characterized by just excessive Staph aureus. So Staph aureus triggers breakdown of the epidermal layer, causes immune dysregulation, things like that. And we know that there are certain strains of coag-negative staph, specifically Staph hominis. There's subtypes of Staph hominis that just naturally kill MSSA and MRSA. So the dermatologists are doing this. They're looking at topically applying these strains of Staph hominis, which are naturally occurring in the environment. onto people with eczema, and they're in phase 2 right now. And this is, and again to decrease Staph aureus burden. And so that's all to say, why couldn't we do this with these knockout bacteria? This is a this is just like, I think the dream potentially is um, potentially applying these knockout bacteria to people as a cream or something, and it could be kind of a longer term repellent strategy that could work pretty well. Um, the last pillar of mosquito control that I want to talk about today is biological control. So we've talked about, what have we talked about? We've talked about chemical, barrier, environmental, and genetic, and this is the last one that I want to discuss. So one big part of biological control has to do with leveraging natural enemies of the mosquito, particularly mosquito larvae. So there are carnivorous insects like dragonflies and damselflies that are often deployed, where you create an environment with foliage that attracts these carnivorous insects, and they eat these mosquito populations, or the larvae at least. Certain fish are also known to eat mosquito larvae. So if you introduce these into these well-characterized breeding grounds, they will eat mosquito larvae. So this is the mosquito fish, but guppies and tilapia also like mosquitoes a lot. I think this is the last big topic, but Wolbachia is a really, really important and different avenue of biological control. So Wolbachia is a gram-negative intracellular bacteria. It's these, like, you can see them in this picture. It's the three kind of spherical looking like structures in an insect cell. So these are gram-negative intracellular bacteria, which are commonly seen in arthropods and nematodes. They're seen in some mosquitoes, but not naturally in Aedes aegypti. And they are maternally inherited in offspring. And they have a symbiotic relationship. So these bacteria protect these organisms from dying against viruses. So, viruses that kill insects and nematodes. So the question for researchers was, what if we introduce these bacteria into Aedes aegypti, which is the mosquito, primarily responsible for dengue? Could it reduce transmission? Um, so what they did is they injected Wolbachia into early stage embryos. These lines stabilized pretty quickly, and then Wolbachia was transmitted to different future generations. And then these mosquitoes were fed blood infected with dengue virus type 2. And you can see here that they did a quantitative PCR, and the total dengue virus was significantly lower in Aedes aegypti mosquitoes that had been injected with Wolbachia. So that's the right to most like bars, different versions of Wolbachia. And you can see that they basically had very low kind of copies of dengue in them. So this was translated into real life as well. So after Wolbachia infected mosquitoes were released into 3 Colombian cities, there was significantly reduced dengue incidence. So the left side, let me see, the left side y-axis is dengue incidence, x-axis is time, right side y-axis is Wolbachia coverage. So, and then, the light blue is, I wrote a note here. So the light blue is partially treated areas, so at least 60% of the mosquitoes had Wolbachia, and then darker blue were fully treated areas. And you can see that once Wolbachia was introduced, and as they kind of increased in coverage, dengue significantly decreased, and for a very prolonged period of time as well. It's really impressive. So Wolbachia infected Aedes aegypti population have been deployed across the world already. They're in 14 different countries currently. And in multiple RCTs, dengue incidence has decreased by 70 to 90% in these areas. It's best studied in dengue, but it's also been effective with Zika, chikungunya, and yellow fever. In Culex and Anopheles mosquitoes, the wool blockade doesn't they don't reach like high enough densities and don't have high enough egg tropism, so doesn't transmit easily. It's not as nearly as effective to prevent West Nile and malaria. Big advantage here is that these lines are stable for many years. They just continue to persist. And then it's also thought to be environmentally safe because these bacteria are already widespread anyway, and there's no genetic modification involved here. So that was it. Really quick tour through some cool, vector control strategies. So I wanted to briefly summarize. So yes, I mean, mosquito-borne illnesses are on the rise. Uh, It's a global concern. An effective control strategy is multi-pronged. Um, particularly because insecticides are not as effective as they once were, and there's just concerns about off-target toxicity and environmental impacts. But there are some promising new developments, and people are continuing to work on these. So for chemical control, we talked about how auto dissemination over traps allows us to dust mosquitoes up with, insecticides for them to bring to their secret breeding grounds elsewhere. We also talked about how ivermectin reduced mosquito survival and could be given to given to humans, and we'll still see if it's safe in birds, um, but people are actively looking at that. We also talked about how bed nets are now being made with either PPO or another kind of active insecticide to decrease the risk of resistance. People are working on developing chemical-free clothing that works just on barrier effect alone. And then Eve tubes are another important way to protect housing structures. Um, for environmental side of things, we're now encouraging like drip or intermittent irrigation systems rather than flooding systems, even with rice paddies and rice fields. Standing water is not good. Um, for genetic controlled, this is a very exciting area of research. The use of CRISPR-Cas9 to make non-viable females and sterile males and PGSIT could safely reduce mosquito population, and then also just genetically knocking out lactate dehydrogenase in skin bacteria could reduce our ability for mosquitoes to sense humans, which is exciting. Um, and then lastly, we talked about how Wolbachia, which is like a naturally occurring intracellular bacteria, can be introduced into Aedes aegypti populations and reduce incidence of dengue and other arboviruses. And I think that's it. Sorry it's so quick. But if anyone has any questions or comments, please feel free to speak up.
Thanks, Eddie. My first comment is never apologize at the end of a talk, especially if you wrapped up exactly at the right time. So that was great. And very interesting, again, I think invoked a lot of thoughts from different people. I want to return to the ivermectin. I'm sorry, there were questions. So I think the summary, and we just wanted to make sure the ivermectin doesn't prevent mosquitoes from biting you, but it's toxic to the mosquitoes after the bite. Okay. Yeah, and do you want to talk?
Yeah, I don't know. I don't know if it's a repellent, because I think that's the question. But we at least know it's insecticidal, so it will certainly reduce the survival of mosquitoes. I don't know. I didn't look into whether or not it's repellent. Yeah.
And the disclosure that you had, I'm sorry, did you talk much about that? And I'm
I
curious.
just mentioned briefly that we have treated fabrics that are insecticide free, and we have a patent on them.
And what kind of fabrics are they? 'Cause I think Gigi wants, like, what type of yeah, what is it all?
They're technical fabrics, so they're designed like they're performance or technical fabrics that kind of already exist but have never been used for this indication. Um, so they're kind of on the lines of something like, they're not like cotton, but they're like a nice polyester that are also comfortable.
It's clothing as opposed to like bedding
Yeah, it's clothing.
or
It's clothing. We've done comfort testing on them as well, so we know that they're comfortable, and our hope is that people would use them and mosquitoes that way.
I think there are a lot of people here who would like to try to wear some of those clothes.
I know.
Nettie,
Well, I'll let you guys know.
yeah. Um, Nettie has a few questions. Nettie, I don't know if you'd
Yeah,
like
I'll
to
just
unmute.
ask him. So,
Yeah.
first of all, thank you so much. That was so interesting. I literally this morning had a text from my sister about travel and mosquitoes. So that was super relevant. I feel kind of itchy, but I am very interested in um, some of these new interventions. So one I'm curious about is the skin microbiome part, and whether there's any sort of lay person suggestions that the dermatologists have for sort of making adjustments to the skin microbiome, just sort of with that notion that, how there's some people that mosquitoes just seem to love, and you wonder maybe that's why. And so I remember as a kid being told, like, take garlic, take this, take that. So any suggestions from the dermatologist about how to adjust our skin microbiome in a positive way? And then I'm also wondering if we're going to get any of these control measures in San Diego, where the mosquito population has shifted over the last couple of years, and now you get bitten during the day.
Yeah, some great questions. I honestly don't know about the kind of like about garlic intake or other other like easier things to do to adjust our microbiome. We already have like skin probiotics, so that kind of decrease odors. We use live bacteria to decrease odors. I don't know if those have been looked at in this respect. As far as in San Diego itself, I think people are looking at the bird ivermectin situation. So that's how I heard about it, is through the Akbari lab, is that there are some nearby labs that are looking at that. So yeah, as far as
Great.
I don't know if Wolbachia is being, I don't think that's being utilized in the US yet.
And any of it, tell them they can come to my house. Thank you so much. That
Yeah,
was
yeah.
very interesting.
And Eddie, was the staph hominis that you talked about that right before the Wolbachia trial?
Yeah.
Do you think one's more promising than the other?
So I
I'm
think
gonna say
this,
just because there's
yeah, so I think the thing about the skin commensals is we can reduce the incidence of, or we can reduce attraction of all mosquitoes, but it seems like the Wolbachia is specifically at this point just with Aedes and not with Anopheles or Culex. So I think but there's been a huge disruption in funding for that project right now for obvious reasons. So I think it's at a huge standstill. And Wolbachia has kind of already been employed before, so easier to fund, I think.
Sorry, this is Sarah Brown. I'm in the car, so you can't see me. But can I ask you, Ed? I just want to give you really excellent, excellent talk. Thank you very much.
Oh yeah, of course,
Um,
thank you.
really impressive, great overview. I really want to give you, please keep going. So, what do you do yourself? We know that the mosquito population in San Diego is increasing, and the number of diseases we're getting, the incidence of the diseases we're getting from those mosquitoes is increasing. What do you do yourself from all the knowledge you've got?
That's a great question. I'd be interested to hear what other people do. Um, I mean, right now I rely on barrier method, like just really good clothing and DEET, which I didn't really talk about here, but there are some limitations to DEET. But I think that's part of it. Like I haven't really traveled to heavily mosquito endemic areas before, except for Alaska, which doesn't have these kinds of mosquitoes. So I'm not you know, practically, like I don't really know the best way to go about doing this. I don't know if I have other people have some insight to how does they manage this. So
No, but it's excellent. Thank you very much. And it's interesting
yeah,
what you're pointing out, that the most likely solution is going to be a combination of methods.
yeah, exactly, exactly.
Thank you so much.
I
Dr. Grillotti.
yes, thank you for calling on me, Jill. So a great talk, actually. I thought it was fantastic. And I actually did some global health before I came to UCSD. So I was under a mosquito net. And honestly, I could understand why there wasn't great uptake, because it is just so uncomfortably, unbearably hot in most areas when you're under a mosquito net. And, you don't have electricity, air conditioning, all that kind of stuff. So in any event, it's very interesting to me now to hear that the mosquitoes have adapted to get around those, or to work around those nets.
Yeah,
But remind me of or kind of made me relate your comment about the comfort of the clothing that that you guys have kind of developed, and the importance of really testing that in environments where they're going to be of most benefit. Because you just have somebody, I mean, I don't know how you actually establish whether or not people thought it was comfortable, but just having people try it on for a few minutes like you would in a store does not necessarily mean that in like real world settings, it's going to be, felt that it's comfortable and light and, not, bothersome, and something that people will want to continue to wear. So just a comment more than a question, and wish you further success in that endeavor.
thank you, Dr. Grillotti. I think we had a tough time trying to measure comfort in our studies. There's some, s- like blind subjective hand-filled tests that we do on a Likert scale of 0 to 10 on the 10 different factors that are associated with comfort. That's what we tried. But the test is actually making the clothing and having people wear it and see if they like it, not just a hand in a bag to see if it generally feels comfortable or not. So I definitely see your point. I think I just had to mention the patent, but like I think we're far from that still. It's promising, but we'll see if people actually like to wear it and if it could practically be of use. Um, it could. The goal is to get the clothing into areas that are plagued by these mosquitoes, but they could also be used in mosquito labs in San Diego. I mean, they're not at risk of mosquito-borne illnesses in the UCSD labs, but could prevent biting. It could be used in other areas as well. So, and then it
Yeah, you
looks
said
like
Anik popped in. Oh, was there something else? I'm so sorry. Were you
no,
just,
I'm looking at the meeting chat about
oh,
yeah, these repellents, Cardin, which is a repellent.
yeah, is
Is
this
that
yeah, did you know about this, Eddie?
I know. Yes, I
Brazilian
do know about
would
this.
know
I
it
didn't know
the
that I
best.
didn't know this was the one that people recommended, but the Cardin is like well known. So
Okay. I mean, the big Goomba question, of course, is that these are on the rise because of climate change, right? I mean, I don't want to simplify
yeah,
it that much. Um, are there efforts to sort of combine this work along with climate change? I don't know if,
yeah.
climate
I
change work.
I didn't look into it, but there's got to be, right? I mean, I think that's a huge motivation to stop climate change, is that malaria is rising and stuff. So
Yeah. Okay.
yeah,
Well, thank you so much, Eddie. Is this oh my God, I keep trying to make you, I'm like, he's an attending now. So
he's done. He's
he's
done.
done. No, we'll
Yeah,
get to hear more about this next year.
yeah.
Hopefully, um, I think that it was really nice to have a different talk, and it was, I agree with everyone, very well delivered. So I hope we can have you back next year to, hear some updates, and anything that you're doing that you can talk about would be so wonderful.
Yeah, thank you guys all for having me and for listening. I really appreciate it. And thanks for the comments and questions.
All right, have a great day and weekend, everyone. We'll see you next week. Bye-bye.